Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Nearly 20 years ago, scientists developed ways to stimulate or silence neurons by shining light on them. This technique, known as optogenetics, allows researchers to discover the functions of specific neurons and how they communicate with other neurons to form circuits. Building on that technique, MIT and Harvard University researchers have now devised a way to achieve longer-term changes in neuron activity. With their new strategy, they can use light exposure to change the electrical capacitance of the neurons' membranes, which alters their excitability, how strongly or weakly they respond to electrical and physiological signals. This new tool is designed to tune neuron excitability up and down in a light-controllable and long-term manner, which will enable scientists to directly establish the causality between the excitability of various neuron types and animal behaviors, says Xiao Wang, the Thomas D. and Virginia Cabot. Future application of our approach in disease models will tell whether fine-tuning neuron excitability could help reset abnormal brain circuits to normal. Wang and Jia Lu, an assistant professor at Harvard School of Engineering and Applied Sciences, are the senior authors of the paper, Which App? When those engineered neurons are exposed to light, changes in the flow of ions through the channels suppresses or boosts neuron activity. By using light, you can either open or close these ion channels, and that in turn will excite or silence the neurons. That allows for a fast response in real time, but it means that if you want to control these neurons, you have to be constantly illuminating them, Sessler says. The MIT and Harvard teams set out to modify the technique so that they could generate longer-lasting changes in excitability, rather than transient activation or suppression of activity. To do that, they focused on altering the capacitance of the cell membrane, which is a key determinant of the membrane's ability to conduct electricity. While many studies have focused on membrane conductivity executed by ion channels, naturally occurring myelination processes suggest that modulating membrane capacitance is another effective way of tuning neuron excitability during brain development, learning, and aging. So, we wondered if we could tune neuron excitability by changing membrane capacitance, Lu says. In that study, published in 2020, Lu used an enzyme called peroxidase to assemble the polymers. However, that approach did not allow for precise control over where the polymers accumulated. It also posed some risk because the reaction requires hydrogen peroxide, which can damage cells. To overcome those limitations, Lu's lab at Harvard teamed up with Wang's MIT lab to try a new approach. Working with neurons grown in a lab dish, the researchers engineered the cells to express this light-sensitive protein, known as mini-SOG. When activated by blue wavelengths of light, mini-SOG produces highly reactive molecules called reactive oxygen species. After several minutes of light exposure, the reactive oxygen species spur those building blocks to assemble into either PDAB or PANI. Thanks for watching. Please subscribe my channel for more videos.